So, welcome to your brain. So the first thing that I want you to be aware of when it comes to your brain is that there are three main divisions of the brain. All of this here is the cerebrum. The cerebrum. Just this outer, more superficial portion of the cerebrum is called the cerebral cortex. The cerebral cortex. We also have the cerebellum, which is here. And all of this here in white is the brainstem. Hills are considered gyri. One hill would be a gyrus. Two or more hills would be gyri. The indentations are sulci. Sulci would be plural. Singular is sulcus. A fissure is sometimes used interchangeably with a sulcus. A fissure is technically a deep sulcus. So a couple of landmarks to get us started. Here we have the great longitudinal fissure, sometimes just simply called the longitudinal fissure. I'll accept either one as a correct answer. Okay, the longitudinal fissure is going to separate the right and left cerebral hemispheres. Turning this to the side, we see another fissure. This is the lateral fissure, also known as the Sylvian fissure. Okay, the lateral fissure is going to separate the frontal lobe and parietal lobe from the temporal lobe. So let's get acquainted with where the four lobes of the brain are. So each hemisphere has four lobes. This one has a Roman numeral number 1 right here, but all of this from here to here, all of this is frontal lobe. We have another sulcus here called the central sulcus. The central sulcus is going to separate the frontal lobe from the parietal lobe. So the central sulcus is right here. And just so you can see what it looks like from the top, so you can more easily identify it, it is here. On this model, there's a number 5 with a little arrow that points to it. So the frontal lobe is responsible for higher order thinking, good judgment, being able to foresee the consequences of one's actions, much of our personality, much of what we think of that makes humans human is located in the frontal lobe. In addition, there is a gyrus right here, and this is the primary motor cortex. The primary motor cortex. So conscious, willful movement is going to be initiated in the brain here at the primary motor cortex. Right. It also has the name because it sits just before or just anterior to the central sulcus. It's also sometimes referred to as the precentral gyrus. Precentral gyrus. Okay, so here I have that central sulcus again, and behind it from here to about here is the parietal lobe. The parietal lobe. Now the parietal lobe has a structure right here, and this is the primary somatosensory cortex, sometimes referred to as the postcentral gyrus, the primary somatosensory cortex. Somato meaning body or conscious awareness of the body, and the sensory information coming from the body will go here to that primary somatosensory cortex. So the parietal lobe, this is your internal GPS or spatial awareness. So this portion here is the occipital lobe. The occipital lobe has the primary visual cortex, and this is where visual information is ultimately processed. So here is that lateral or Sylvian fissure again, and inferior to that I have the temporal lobe or temporal lobe, however you wish to pronounce it. So the temporal lobe is responsible for some memory processing and for auditory processing. Okay, auditory processing. The cerebellum is responsible for balance and coordination. If I damage the cerebellum, I'm going to have a difficult time walking, catching a ball. Walking a straight line is going to be extremely difficult for me. So I'm going to kind of revert to how I walked maybe as a 2-year-old. So also hand-eye coordination, that sort of thing. Now all of this is brainstem, but we have different areas of the brainstem that you need to know. Here, this part is the mesencephalon or midbrain. The mesencephalon or midbrain. Here where we have this bump, this is the pons. Inferior to the pons, we have the medulla or the medulla oblongata. All three of those together make up the brainstem. One of the things I don't have on here is the pituitary gland. For those of you who do have a pituitary gland, it's going to sit right here, and it's going to be connected just by this little hook, and it looks like a grape. So that's the pituitary gland. And you might remember the pituitary gland actually sits in the sella turcica of the sphenoid. That's responsible for secreting human growth hormone. Okay, a tumor in this area that causes an excess of human growth hormone causes acromegaly and gigantism, growing very, very large. Usually have a very short lifespan because the heart can't keep up with the size. Here's a Roman numeral 1. Okay, this is cranial nerve 1. Cranial nerve 1 is the olfactory nerve. The olfactory nerve processes your sense of smell, olfactory information or sense of smell. The axons of the olfactory nerve go through this area here at 8 called the olfactory bulb. The olfactory bulb. And those axons also are going to go through the cribriform plate that you might recall from the ethmoid bone of the skull. All right, I also have a Roman numeral 2 here. Cranial nerve 2 is the optic nerve. The optic nerve. So the optic nerve is formed by the axons of ganglion cells coming from the retina. The axons are going to bundle together to form the optic nerve. They then flip a U-turn and exit the eye at the optic disc. We'll go over that when we look at the eye. From the point where it exits the eye, it's going to go right here. Just after this point, it's going to cross to the other side of the brain at this structure here that kind of forms an X. This is called the optic chiasm. The optic chiasm. Now I have these 2 little bumps here. So these are called the mammillary bodies. And these actually have an important role in declarative memory or episodic memory, such as telling me what happened on your way home from school yesterday or whatever. Just this outer portion here is the cerebral cortex. The cerebral cortex. The whole thing is the cerebrum. This portion is the cerebellum, and all of this is brainstem. This structure here, where it says 10, that is the corpus callosum. The corpus callosum is the connection that exists between the right and left hemispheres. The corpus callosum. Underneath the corpus callosum, if I put a sticker here and ask you for the space, okay, there is a fluid-filled space. The space is filled with cerebrospinal fluid, and this is actually a lateral ventricle. A lateral ventricle. Now, there's a lateral ventricle in the left hemisphere and a lateral ventricle in the right hemisphere. And they kind of take the place of lateral ventricles 1 and 2 because we don't have a 1 and 2. Now, if I put a sticker here and I ask you for the structure that is here is the septum pellucidum. Now, the septum pellucidum is a physical separation that separates the right and left lateral ventricles from one another. Since we're on ventricles, let's keep with that for a moment. So here is that lateral ventricle. Where number 20 is, that is the third ventricle. The third ventricle. Now the third ventricle has a structure here at 26. Now this structure is called the choroid plexus. The choroid plexus. The choroid plexus has ependymal cells, and they are the ones that are going to create cerebrospinal fluid. All right, and it's part of the third ventricle, which is denoted here at number 20. From the third ventricle, cerebrospinal fluid is going to go through this area here. It's denoted as number 34 with a little arrow, but just so you know, it's all of this. Okay, that is the cerebral aqueduct. The cerebral aqueduct, also called the aqueduct of the midbrain. And here, just between the brainstem and the cerebellum here, I have the fourth ventricle. The fourth ventricle. And all of the ventricles are filled with cerebrospinal fluid, the same cerebrospinal fluid that's going to bathe the spinal cord. The floor of the lateral ventricles is made by a structure called the fornix. The fornix. This structure here is part of the third ventricle, and remember this is the choroid plexus, has ependymal cells, makes cerebrospinal fluid. This little point here is the anterior commissure. When you hear the word commissure in neuroscience, it just means connection. Okay, it's like a bundle of fibers. So this is the anterior commissure, and here at I think it says 13, this little thing here, that would be the posterior commissure. Okay, so these are additional connections that connect the right and left hemispheres in addition to the corpus callosum. Here at 19, this is the thalamus. The thalamus is the sensory relay center of the brain. So all of your senses, except for smell, are going to go to the thalamus before they get relayed to the higher centers of the brain for actual perception. Below that, here at 21, we have the hypothalamus. The hypothalamus. The hypothalamus does a lot of stuff, including sleep, wakefulness, alertness, hormone control, appetite. Now, there's a connection between the thalamus and the hypothalamus, and that connection is right here at 22, I think. Here is the interthalamic adhesion. It's going to allow communication between the thalamus and hypothalamus. Okay, interthalamic adhesion. Adhesion, just think of adhesive, glue. Here at number 23, this is the pineal gland or pineal gland, whichever way you wish to pronounce it. The pineal gland is going to secrete melatonin and make you feel sleepy when it's dark outside. Okay, so the pineal gland is what dictates your sleep-wake cycle. Right here, this whole thing is called the tectum, okay, at 31. There are two bumps here of the tectum. This top one here is the superior colliculus. The superior colliculus is responsible for visual reflexes. Let's say that you see something large moving in your peripheral vision, or you see a bright flash of light. The superior colliculus is going to orient your head and eyes toward that stimulus. Okay, so defensive mechanism. The lower bump here is the inferior colliculus. The inferior colliculus does the auditory reflexes. If you hear a loud, startling noise, this is the center of your brain which is going to automatically orient your head and eyes toward the location of that sound. This is cranial nerve 2 here. That's part of the optic nerve. This is a mammillary body that's here. This whole area here is considered the midbrain or mesencephalon. Where we see this is the pons, and inferior to the pons, again, we have the medulla or medulla oblongata. Now, this whole structure here is the cerebellum. These white branches, the white matter inside of the cerebellum is called the arbor vitae. The arbor vitae. And that's everything you need to know for the brain. Any questions before we move on? This is your eyeball. So first of all of this white area here, all of this is sclera. So sclera is the white of the eyes, sclera. Now, if I were to pop your eye out of your head, you would see that the sclera actually continues, and it even continues towards the back of the eye. So all of this is also considered sclera, all of this white here, until we get to this portion here, which is the optic nerve. Okay, so this is the optic nerve. So this is the superior rectus. Remember, rectus means straight. Superior rectus allows the eye to gaze upwards. I have a diagonally oriented muscle here, which is the superior oblique. Oblique meaning diagonal. All right, so superior oblique. What you want to remember is that the superior oblique is going to point toward the medial side. Okay, superior oblique points towards the medial side, making these muscles the medial rectus. Medial rectus allows the eye to gaze toward the nose. Medial rectus. Around. Okay, so here we've got the superior rectus, superior oblique, medial rectus, and then if I turn this around, here I have the inferior oblique, and the inferior rectus. Inferior rectus allows you to look downward, and this would mean that this is the lateral rectus. The lateral rectus allows the eye to gaze outward from the nose, so away from the nose, or abduct the eye. In this model, I have a portion of the sclera cut away. The outermost layer of the eye is the sclera, again, the sclera. The innermost layer of the eye is the retina. Okay, so this orange here is the retina. The layer in between the retina and the sclera is here, and this is called the choroid coat. The choroid coat. The choroid coat has the arteries and veins that are going to feed the very hungry retina. So the choroid coat is going to provide that blood supply. In addition, it's going to wash away waste products and carbon dioxide. This outermost portion here is the cornea. This is the cornea. All right, if you get corrective eye surgery, they can actually change the shape of the cornea to assist your vision. All right, so this is the cornea. All right, it's convex. And I'm going to put that on the side because I could ask for it just on the table too. All right, so remember that. After the cornea, I have the iris. Okay, and just so you know, there's a chamber between the cornea and the iris. All right, so this chamber right here. This chamber would hold the aqueous humor. The aqueous humor. And let's look at the iris in a little bit more detail. The colored part of the eye is the iris. The iris, and that is here. The iris acts like an aperture of an old camera, and what that does is when the iris contracts, the hole here is going to get smaller. Now the hole here in the iris is actually the pupil. So the pupil is not a structure that you can hold in your hand. The pupil is a hole, and it's a hole in the iris. The size of that hole is governed by the iris itself. All right. When the iris relaxes, the pupil gets larger and more light is allowed to enter the eye. That's called dilation. Okay, so When you go to the doctor, one of the things that they might do is they might shine a light in your eye. When they shine the light in your eye, one of the things they're doing is they're looking for a pupillary reflex. When I shine a light in somebody's eye, the iris should constrict and close, making the pupil smaller. Okay. When the pupil constricts, less light is able to enter the eye. Beyond the iris, if we remove the iris, the next structure that we see is the lens of the eye. So this is the lens, and the lens is a biconvex structure, like a spaceship. Okay, biconvex, it's convex on both sides, whereas the cornea is convex only on one side, right? So it's just convex, that's it. This is biconvex. I'll try to show you better. Okay. What the lens does is the lens allows for accommodation. In other words, it changes shape when I want to look at something close versus far. If I'm looking at something far away, my lens is flatter. When I look at something up close, my lens is actually going to change shape to become more round. And the reason for that is because it's focusing that image onto the retina all the way back here. Before I go too much further, so just like we have the aqueous humor that's in the chamber between the iris and the cornea, we also have this larger chamber of the eye here that is fluid filled. So this is representative of that fluid, okay, in the globe of the eye. So this is called the vitreous humor. The vitreous humor. Okay, so all this is vitreous humor. That is the liquid or gelatin-like substance that is in the eye that helps maintain eye pressure. When you go to the eye doctor, sometimes they'll do a little puff of air. And what they're doing there is they're actually testing, they're measuring the pressure of your eye, the intraocular pressure. When you have health problems in your body, a lot of times it will show up in places like the pressure of the eye or the back of the retina. Things such as stroke or being close to a stroke or having very high intracranial pressure, those types of things can be shown not only in the intraocular pressure, but also in the appearance of the retina. When a doctor is looking with an ophthalmoscope through the pupil, they're actually seeing the retina. Now, I mentioned that the lens changes shape depending on whether you're focusing on something close or far, but I didn't say how. So let's talk about that next. We have the ciliary body, which is denoted here, okay, or represented here by this brown ring. Okay, this is the ciliary body. The ciliary body are muscles that connect to the lens via suspensory ligaments. All right, so the suspensory ligaments are here in this model. And that's what controls the shape of the lens. As a person gets older, they have a harder time focusing on stuff that's close, because when things are far away, this is relaxed and the muscles are relaxed. It's flatter. But when I try to look at something close, the muscles of the ciliary body are going to constrict. The suspensory ligaments are going to pull on the lens, making it rounder. Okay, so that activity gets weaker as you age. So a lot of times when you hit 45, 50 or so, you might start needing glasses, and maybe you never needed them before. And it's because we start losing that close-up accommodation, because these muscles just get tired with old age, unfortunately. So on the outside, I've got the white, which is the sclera. And then I have this red here. This red is that choroid coat. Okay, remember the choroid coat has all the blood vessels that are feeding the retina. Okay, in orange here, all of this area here, that is retina. All of that is retina. The sharpest point of your vision is right here. There's a little yellow dot. I also have the other half of the dot is on the other side of the model. This is the fovea or fovea centralis. The fovea or fovea centralis. If I go out a little bit from that, this whole area would be called the macula. You may have heard of macular degeneration. All right, so the fovea centralis is the center point of the macula. So it's going to be the center point of your vision that is most crisp and clear. Okay, we've got two types of photoreceptors. We've got rods, which are responsible for black and white, and cones that are responsible for color. The reason why we have such crisp, clear vision at the fovea or fovea centralis is because this has the highest concentration of cones in the retina. As I get more and more peripheral or more into my peripheral vision, I actually see less color and more black and white. That is good for seeing motion. Okay, so we can see motion. We might not be able to distinguish what it was that moved, but we are able to detect motion in our peripheral vision a lot better because we have more rods than cones, and it helps us see that contrast. Also, the rods are really beneficial for low light situations. So this is the optic nerve. The point at which the optic nerve leaves the eye is called the optic disc. So this is the optic disc here. There are no photoreceptors here. There's no rods or cones. The portion of my vision that lies on the optic disc is called the blind spot. Okay, so you actually have a blind spot in your vision that you're not aware of. And this is what creates that, because there are no rods and cones at that little point of the optic disc. The ear is separated into three regions. From here outward is the outer ear, okay, or external ear. The middle ear goes from here to here, but also includes this structure. That's the middle ear. And the internal ear, or inner ear, is going to be these structures here. All right, so let's start with the outer ear. The outer ear has this structure here. This is known as the pinna or the auricle. The pinna or the auricle. The reason that this has this unique shape is this helps the brain to distinguish the vertical position of a sound. So I can tell whether a sound is coming from above me or below me because this is asymmetric. In order for me to tell whether a sound is coming from the right or left, the brain actually does a little mathematical calculation and can detect that slight disparity, the slight difference in whether the sound reaches the left ear first or the right ear first. Okay, so it uses all of that together so that you can locate in three-dimensional space the direction that a sound came from, so it's pretty cool. Another thing this does is this allows for funneling the sound waves into the external auditory canal, which is here, the external auditory canal. The external auditory canal is going to end at this structure here. This is the tympanic membrane. The tympanic membrane, colloquially called the eardrum. So once we get to the tympanic membrane, at that point we are considered to no longer be at the outer ear, we're now at the middle ear. The structures of the middle ear are going to include the tympanic membrane and the three ear ossicles. Ossicle means tiny bones, and you have three of them. You've got the malleus, incus, and stapes. In a practical exam, it's very likely for a practical exam this may be unarticulated. It might just be sitting on the counter like so. So this is the tympanic membrane. Attached to the tympanic membrane is the malleus, and then the incus. Okay, so the malleus is first. Malleus, the incus, and in this model, the stapes is actually connected to the vestibule of the inner ear. So this is the stapes. Okay, malleus, incus, and stapes. So when the tympanic membrane is vibrating, it's vibrating just from the sound waves of the air, kind of like a speaker would be doing, especially if the speaker had a lot of bass. You can really feel those vibrations. Well, the vibrations that are traveling through the air are relatively weak. So you could imagine if I'm running through the air, it's a lot easier than me trying to run through a swimming pool of water that's 5 feet deep. What the malleus, incus, and stapes really do is they're going to amplify that sound. So as this tympanic membrane is vibrating, all 3 of these ossicles are going to be moving along with those vibrations. And they use a trick of physics and combining levers together to actually increase that signal. So much so that once I get to the stapes, so this is going to have enough pressure to actually create a liquid wave within these structures here. I need to have enough pressure to actually move at the oval window, move that enough so I can move liquid and create waves within these structures. Okay. One last thing you need to know about the middle ear is we have this long structure here. This structure, sometimes called the auditory tube, I don't like that term as much because it has nothing to do with hearing. The other term that I like better is the eustachian tube. The eustachian tube. The eustachian tube equalizes pressure between the middle ear and the outside world. So now let's go to the inner ear or internal ear. The internal ear is going to start not at the stapes, even though it's like connected there, but it's going to start at the oval window. Now I'm going to take this off so we can see these structures better. So here is that stapes. Here is the stapes. Okay. The stapes is connected to the oval window. The oval window is right here at the bottom of the stapes. This entire area, all of this, all of that, all of this, even though the number is, even though the number for this is right here, I could put the sticker anywhere. All right, so all of this area down here is the vestibule. The vestibule. Okay. I have 3 semicircular canals that are going to go up here. There's 1, 2, and 3. All right, these are oriented in the 3 planes of movement, X, Y, and Z. At the base of each of these semicircular canals, we get an ampule. Okay, so each one of these is an ampule or ampulla. All right, let's see. So this whole structure here is the cochlea. The cochlea. Now the cochlea is filled with hair cells that sense sound information. If I were to unwind the cochlea, it's set up like a piano. Interestingly, sound waves must first go, so it starts at the high end of the piano though. Okay, so every time you hear a sound, the sound waves have to travel through all of those high notes to get to the correct note, and then those hair cells are going to vibrate. Okay, and that's how you hear the pitch of a sound. But because of the way the cochlea is set up, this is why as you age, you actually start to lose the higher pitches of sound. The reason is the hair cells that are the higher pitches are at the base, and they get fatigued with age. Okay, isn't that interesting? If it was the other way around and the bass came first, we'd lose the bass first instead of the treble. Yeah, instead of the high notes. Kind of interesting. Okay, so the cochlea information is going to travel through this here, which is the cochlear nerve. Now the cochlear nerve and vestibular nerve come together to make the vestibulocochlear nerve. Okay, so here we have the auricle or the pinna. Here is the external auditory canal. The external auditory canal ends at the tympanic membrane, which is here. The tympanic membrane is connected to the malleus, which is here. The malleus is connected to the incus, which is here. And the incus connects to the stapes, which is here. Okay. The stapes is connected to the oval window, which is located on the vestibule, which is this bottom portion here. Before I forget, this is the eustachian tube, sometimes called auditory tube, eustachian tube. All right. So this down here is the vestibule. Each of these is a semicircular canal. Semicircular canal. This is the cochlea. This is the vestibulocochlear nerve. The vestibulocochlear nerve is here. And at the base of each of the semicircular canals, we have the ampullae, or ampulla. What this is this is actually C5. Okay, this is the 5th cervical vertebrae here. And this is showing you the vertebrae as well as the epidural space. This is where you would get an epidural. You also see the meninges, the 3 coverings of the brain as well as the spinal cord. And then this here is a cross section of the spinal cord. So this being a cervical vertebrae has the transverse foramina. Oops, the transverse foramina. And they're here and here. The transverse foramina are the passageway for the vertebral arteries and vertebral veins. Out to the side here, and I have one on the other side too, right there. Okay, these are the transverse processes. Transverse processes. This here is the spinous process. The spinous process. These are the superior articulating processes, here and here. And this is the body. And remember, we have the body, disc, okay, so on and so forth. Let's go over some of the coverings and structures you see here. So this is the epidural space. Furthest out, we have the covering or meninges called the dura mater. The dura mater. Deep to the dura mater, we're going to have the arachnoid mater. Arachnoid mater. The arachnoid mater actually is called that because it actually has structures that looks like a spider web. And below that, we actually have the subarachnoid space. The subarachnoid space. Now, this space is filled with cerebrospinal fluid. Deep to that, we have one more, the 3rd covering, which goes just on the outside of this cross section of the spinal cord. Okay. And that is the pia mater. The pia mater. So the pia mater is the deepest one. The first structure I want to point out is this little hole here in just the center. This is called the central canal. The central canal. The central canal is filled with cerebrospinal fluid, just like the subarachnoid space. All of this here is considered white matter. And in the middle here, this is all considered gray matter. There's a couple landmarks that I'd like for you to know. So this is the posterior portion and the anterior portion. So that's going to make this fissure here the anterior median fissure. The anterior median fissure. In addition to that, I also have dorsal or posterior median sulcus. So this portion here of the white matter is called the anterior column. Okay, the anterior column. You can also call this the ventral column. the ventral column. And this is the posterior or dorsal column, this white matter here. Here, laterally, is, as you might guess, the lateral column, okay, of white matter here. This is the central canal again, and around it, these straight things here, this is the gray commissure. Okay, commissure meaning like a connection. Okay, so this is the gray commissure. These here and here, this would be the ventral horns, and here would be the dorsal horns. Dorsally is where we have sensory information. Ventral, the ventral root is here at 17, because this is on the ventral side. This is the dorsal root, and this bulge here is the dorsal root ganglia. Dorsal root ganglia. Okay, 17 has motor information, and that's the ventral root. So in this model, so this is all the white matter here and here. Gray matter is here and here. The central canal is located right here. Okay, central canal filled with cerebrospinal fluid. This is the anterior portion and the posterior portion. This is the anterior median fissure here, and the posterior median fissure or posterior median sulci. This is the lateral column. So this is lateral column and this is lateral column. This portion here and here, this is dorsal column here and here, or posterior column. And this is anterior or ventral column here. This is the dorsal horn. This is the dorsal horn. This is the lateral horn, and the ventral horn. Ventral horn. This is the ventral root. This is the dorsal root. And this is the dorsal root ganglia. This is the dorsal root ganglia, and the ventral root on this side. When the ventral and dorsal roots come together, at this point, we call this a spinal nerve. Okay, and this is at that point where the spinal nerves actually exit the vertebral column through the intervertebral foramina. Okay, any questions about that?